everyone. In this video we will be seeing about hemerythrin and hemocyanin. If you really want to know how some animals are having blue colored blood or even violet or pink colored blood, then this is for you. So this is mainly because of the oxygen carriers hemerythrin and hemocyanin. So initially we will be seeing regarding the deoxyforms, later we will go on to the oxyforms. So hemerythrin. So this is mainly found in marine invertebrates. So what are invertebrates? So invertebrates are animals that doesn't contain any backbone or there is no internal skeleton. So if you want to know the many properties of invertebrates, you can search in Google and learn about it. So I have mentioned some examples: sea spider, arrow crab and brine shrimps. There are many other examples too. So these are some of the examples of marine invertebrates. So this is a non-heme protein. What does it mean by non-heme protein? Means it doesn't contain any heme group. So what is a heme group? Heme group is a metalloporphyrin with many substituents. So there are some of the heme protein, that is example hemoglobin, as we have in our body as an oxygen carrier. But this is a non-heme protein which doesn't contain any heme group. So their main function is to transport oxygen, and the oxygen is transported reversibly. That is the process of this into deoxyform and converting into their oxyform is a reversible process. So how is the overall structure of this looks like is, it contains around 8 subunits, and in each subunit it has 2 ion centers and it is surrounded by the protein part. And the protein part here is made up of around 113 amino acids. That is, each subunit is having 2 ion centers and the protein part is made up of 113 amino acids. Coming to deoxyhemerythrin. So this is the structure of deoxyhemerythrin. As you can see, it contains 2 ion centers. The 2 ion are in their +2 oxidation state. So oxidation states are very important. These are asked in many exams. Oxidation states, in what type of spin state they are in and what is the arrangement around each metal. These are some of the familiar questions. So these 2 ion atoms are bridged by 3 bridges. So 2 of the bridges are carboxylator bridges and one of them are oxo bridge. So this carboxylate bridge is formed from the glutamate and aspartate amino acids. So this is the structure of aspartic acid and this is the structure of glutamic acid. Here the COOH group is present. So this COO group, the COO group present here is bridging, is acting as a bridging group between these 2 ion atoms. And here this is the mu-oxo bridge. So this mu-oxo group can be either from water or from OH minus or even from O2 minus, but mostly it is from OH minus. And as you can see here, this Fe is surrounded by 6 bonds, that is, it is having an octahedron arrangement. But here this Fe is only surrounded by 5 bonds, thus it is having a 5 coordination around them, and there is one vacant site present there. And in the case of here there is 3 histidine groups, but here there is only 2 histidine groups. And there is antiferromagnetic coupling between these 2 Fe atoms, that is, this FeO Fe bridge is having an antiferromagnetic coupling. So antiferromagnetic coupling is mainly happening between 2 atoms that are in their high spin state. So here these 2 atoms are in their high spin state. This was confirmed using Mossbaer and EPO spectroscopy. So the structure, the center, we can name it as mu hydroxo. That is, what is this mu? This mu is represent the bridged position while we are naming an inorganic compound. So mu hydroxo, bis means 2. Bis carboxylato diion 2 center. So this is to name the center of them. So here there is one vacant terminal coordination site in one of the ion. So So the entire molecule is colorless in this deoxyhemerythrin form. Coming to the oxyhemerythrin form, this oxyhemerythrin form is violet pink in color. So here the oxygen is bound in the vacant site, that is in the 6th position, which was vacant earlier in deoxy form. Here it is occupied by the hydroperoxy group. How we can see it? Initially this Fe will donate 1 electron and this Fe will donate 1 electron. So there is loss of oxygen. So this process is oxidation, and since they have donated 1 electron, it will be converting into Fe3+. So to whom they are donating? They are donating it to molecular oxygen. That is, the molecular oxygen is initially containing 12 electrons, that is O2. Later they have accepted 2 electrons and it will be converted to O2 2-, that is, now it is containing 14 electrons, that is peroxide form. So initially the peroxide form is bound to the 6th position. Later on what happens is this mu oxo group hydrogen will shift from here to this peroxide group, so that it will form a hydroperoxy group, that is HO2-. So the hydroperoxy group is the ligand that is present in the 6th state. Later on, this hydrogen and the oxygen group present in this bridge position will form a hydrogen bond. So here this oxo group acts as the acceptor and this hydrogen group will act as a donor. And similarly, as I have mentioned in the deoxy form, there is antiferromagnetic coupling between these two Fe3+ state metal atoms, and hence they are in the diamagnetic ground state. Okay. So this is the overall process. See, they are reversible. Here it is very clear there is 6 bonds now in the oxy form. The oxygen is bound and that is the hydroperoxy group is bound, and there is a hydrogen bond between the oxo group and the hydrogen in the hydroperoxy group. So how do we confirm that this oxygen is present in the peroxide form means? This was confirmed using Raman spectra. So these are the standard values for various species of oxygen that was recorded. So how these values are varying means based on their bond strength. So if the bond strength is high, the stretching frequency is high, and the bond strength is directly proportional to bond length, that is inversely proportional to bond length. So if the bond strength is high, then the distance between the bond is very low. Consider for example single bond and double bond. The double bond is very strong, but the distance between the bond is very low. As you can see here the bond strength is high between OO in this O2+ species. That's why the stretching frequency is also high, but the distance between them is very low. But consider this peroxide, there this bond strength is very weak. That's why the stretching frequency is also weak, but the distance between them is very high. This denotes the strength is very weak. And if we now we have recorded this Raman spectra for our oxyhemerythrin, and the value was found to be 844 centimeter, and this was in close relation with the free O2 2- value. That's why we have confirmed that the oxygen is in the peroxide form. So next, coming to the hemocyanin. So hemerythrin will give violet pink colored blood. This hemocyanin will give blue colored blood. So mainly in household, if we say spider, most of the species of spider is has blue colored blood. So this is also found in invertebrates like mollusca, example snails, squid, whales, arthropoda, example crabs, lobster, shrimps. So this is containing a metal called copper in them, that is it contains 2 copper metal, and there are several subunits. So we cannot give a standard value like these many number of subunits are present, because it varies based on maybe this snail is containing 2 number of subunits, but this squid is containing around 8 number of subunits. So we cannot predict it. That's why it is just said several subunits. So the main function is to carry oxygen. So coming to the deoxy form, in the deoxy form the 2 copper is in their +1 oxidation state, and each copper is surrounded by 3 histidine groups, as you can see, and this overall structure is giving a colorless molecule. And see, this is the very zoom in look of this deoxyhemocyanin. Initially the copper is in +1 oxidation state and it is surrounded by the histidine group. Here they have just mentioned the imidazole part of the histidine group, and this pink color is the protein part. So this is the protein part, that is, this protein part will look like a helix, you know the structure of these proteins. And here this is the center, copper center. Here the copper 2 copper is present, that is in their +1 oxidation state. If we zoom in this place, we will have a closer look like this. Here each copper is surrounded by 3 histidine groups. Here just they have mentioned the imidazole part of the histidine group. Coming to oxyhemocyanin, it is containing 2 copper, but they are in the +2 oxidation state. What happens is, this copper will donate 1 electron and this copper will donate 1 electron to the oxygen. That's why they too will get oxidized to copper 2 plus. And now our oxygen, which was initially in the 12 electron state, has gained 2 electrons and it will become peroxide, that is O2 2 minus, and it is getting, that is the molecular oxygen is getting reduced to peroxide. And why is it showing blue color? Means because of the ligand metal charge transfer transition. So this is a very important note you have to take. And these 2 copper can only bind only 1 oxygen. They cannot bind, each cannot bind 1 oxygen, but they both will bind 1 oxygen. And initially we have seen in hemerythrin also. Similarly here also the 2 copper is antiferromagnetically coupled, and hence they are diamagnetic in their room temperature. And how do we prove that this oxygen is in their peroxide form? Means using Raman spectra, and when we calculated the value in Raman spectra, it was found to be 750 centimeter inverse. As you can see, the standard value of O2 2 minus is 802, but here it is little bit low. This shows that the bond strength is even more weak than O2 2 minus free species, because maybe because of the bonds with other metals, this bond between O and O bond is very long, the length is very high, and hence this bond strength is very low. And we can name this center like mu peroxo dicopper 2 center. So that's it for today about blue blood and violet pink blood. So my references are Principles of Bioinorganic Chemistry by Lippert Berg and Organometallic and Bioinorganic Chemistry by Ajay Kumar. Hope you like this video. Thank you for watching. Thank you for watching and supporting me. If you have any doubts and queries, you can mention in comments. I'll reply. Thank you.